person who will moderate this panel is a very good friend of AAKP, Dr. Rebecca Schmidt. She's the first female president of the Renal Physicians Association. She's a recipient of our AAKP Medal of Excellence. She's a champion of care in her state of West Virginia. She's the section chief and professor of medicine with tenure for West Virginia University School of Medicine. She's been a champion all her life, and she has really dedicated herself to improving the lives of folks in a rural state, but taking those lessons and communicating them out for how we can change care for everyone. And I'd like to ask Dr. Schmidt to come on up. And I'd like to ask the guests for the next panel to come on up as well.
Well, good morning, and it's an honor to be here, and like Elena, no greater honor has ever been bestowed on me than to receive the Medical Excellence Award this past spring from the AAKP, so I thank you very, very much. That was a hard interlude to follow, but it means all the more because I was one of the lucky people with Paul, Richard, and Bill at the White House that day, and I think I'm surprised you didn't tell the story, Paul, about at the end of the day, the meeting, Bill took off his nice shirt and tie and paraded around, modeled his kidney, I need a kidney shirt. It was classic, and everyone got a really big kick out of it at the White House. So I guess I'm gonna have to tiptoe up here for this microphone, unless maybe I can do this. So we're gonna talk about innovation. What is innovation? Well, innovation is a lot of things, and we hear a lot about not having had any innovation in the kidney space for a long time, but some of you may remember when we didn't have EPO, so that was an innovation. Did anybody remember that here? Okay, you remember that. And it wasn't pretty, and it wasn't very fun. Some of you may remember that we didn't have the cycler for peritoneal dialysis. My patients now say, well, why do I have to learn how to do it manually? Because your cycler might break down in the night, and then you need to know how to do it manually. We used to only do it that way. So there have been some innovations, and innovations can be all sorts of things: technical, educational, informational, scientific. And you're going to hear a lot of really fantastic things in a few minutes. As a doctor, it's very frustrating to not have innovations, just as it is for you patients. And sometimes, as I've said, all that I can give as a doctor is kindness and a commitment that I'm never going to stop caring, and I'm never going to stop trying to find something to make it better for the patients that I take care of. And the opportunity to be here and for you to see what your organization is doing and how it's dedicated to the future of the patients is really quite an honor. AAKP has encouraged advancement in all sorts of realms, and you've probably going to see this slide again, but their Center of Patient Engagement and Advocacy, it is all about innovation. AAKP is about innovation. And similarly, it's dedicated to innovation in its advocacy and helping you advocate, capitalizing on your strengths and what you know as a patient to make things better for yourselves and the future patients. You, as Elena said so nicely, you are the key, you are the voice, and improving innovation and moving it. Federal agencies need to hear from you and what it's like. And I forget who said it, or maybe it was Paul earlier, but it's when you go to the Hill with one of the patients, the person at the table says, oh, hi, Dr. Schmidt, and what do you have to say? And I say something, then they say, the patient. They want to hear from the patient. They don't really care about what I think. They want to hear from you. So it's really, really true. And the fact that there are 73 of you from 43 states that can help with this type of advocacy is really, really cool. And AAKP has done incredible things reaching out globally to the community. So be proud, be proud. So what is innovation? I think of innovation as having the ideas to inspire us and the inspiration to innovate. But I think Thomas Edison said it really nicely and simply. There's a better way to do it, find it. And that's what we need to do. So with that, I will introduce our speakers. First, we have, where's Diana? I don't have my, is this the right order? We have an order issue. Is this the right order?
Yes.
Okay, all right. So today's speakers will bring not just innovation, but hope. And I think that is the most important thing to take away from this session, is that there is hope. And the first speaker, Dr. Ekemini
You're right. You got it.
Ekemini Riley from the Milken Institute will tell you all about the efforts to develop a philanthropic database and ways to address the unmet need of kidney patients. And we'll start with you, Dr. Riley. Please welcome Dr. Riley to the stage.
Good morning, everyone, and thank you, Rebecca, for the kind introduction. So what I hope to do today is really to infuse hope to you all. So I'm here to really take you all on a journey. One, I want to just tell you a little bit about the Milken Institute, and I'm sure you all are wondering what a think tank is doing at a kidney patient meeting, but I'll be happy to tell you about that. And then tell you a little bit about medical research funding and the role of philanthropy. So my work specifically, I focus on medical research funding and really the intersection of that world and philanthropy. And then I will start the journey with the Giving Smarter program for kidney failure that was kicked off by Rob and Cindy Citrone. And then what evolved into the Play It Forward Pittsburgh campaign for organ donation, and now what we're helping to develop, the Starzl Network for Excellence in Pediatric Transplant, which is a huge unmet need in the space. I am going to summarize nearly 3 years of ongoing work in about 10 minutes, so hold on to your hats. So I'll start by telling you all a little bit about the Milken Institute. So we are a nonprofit, nonpartisan think tank, and our mission really is to increase global prosperity by advancing collaborative solutions that will, one, widen access to capital, create jobs, and improve health. And really where my work lies is in the improving health sector. So our organization has 8 centers, and the work spans across regional economics to health issues and also philanthropy. And what you'll see is that we believe that strategic philanthropy is really at the center of all of these things. And so myself, a scientist by training, I came to this organization to really think critically about how funding can really go towards advancing promising medical solutions to really help patients in need. And so our mission at the Center for Strategic Philanthropy is to improve the strategic deployment of philanthropic capital. When you have capital sources to allocate, I think it's really best to make sure that they're going to the most high value things that they can. And so we really work with our philanthropists and family foundations to really design high-impact solutions for them to create real and lasting change. And we like to say at our organization that medical research philanthropy, you don't go to it, it comes to you. So typically we work with families who have themselves been afflicted by a certain disease or loved ones within the family been afflicted by a certain disease, and they're really thinking about how they can take their capital source and really put it to use in an engaged manner rather than just passive check writing to put their name on a building, so to speak. And so we work, as our center, we work across a number of different focus areas. And I'll be focusing specifically today on advancing medical solutions. That's my area of expertise. So we work with philanthropists across a wide range of areas. So our chairman, he himself is a medical research philanthropist. And he firmly believes that philanthropy is far more than just writing checks. It really takes an entrepreneurial approach that seeks out best practices to empower people to change the world. And so on our staff, we have scientists, change makers, folks who are really looking to take that risk capital, which is philanthropy, to put it to use. And so where does philanthropy fall in this whole ecosystem of medical research funding? So we have our players, we have industry, we have the government at all levels, universities, and then philanthropically funded foundations and independent research institutes and individuals. And as you all, I'm sure, are probably aware, the industry and government make up the bulk of medical research funding, followed by universities with their private endowments. And philanthropy makes up about 5% of the funding for medical research. And so with that small percentage, why philanthropy? And we really believe that philanthropy is far more nimble than other funding sources. It can be patient, it can really take on long-term efforts and also take on risk that other entities might not be able to take on. So while a small source of funding, we do see it as a capital source that can deliver truly transformative and outsized results. And really looking at how it can fill strategic funding gaps that government or industry cannot quite fill. And so, as our executive director always says, we really need to ask how philanthropic funding can catalyze change beyond what's achievable through our conventional funding sources. And with that, we started working with Rob and Cindy Citrone about 3 years ago, and we have seen in working with various philanthropies that when they engage in using the 4 T's of philanthropy, we see much quicker results and much wider results. So we're looking for philanthropists who don't just want to give of their treasures, their financial resources, but also their time, their talent, and their ties. And so that's really what they came to us to try to figure out. How can we take all of these different things and put it to use to catalyze change and advance solutions? So I'll give you the backstory to this. The patriarch of the Citrone family has end-stage renal disease, and he's really looking at what his legacy would be when he passes. And he, still alive now and looking for ways to both better his life and his experience on dialysis, but then also thinking about, what comes after and how can he make a lasting impact on other kidney patients like himself. And so Rob and Cindy Citrone took up that mantle to really try to figure out how they can use their capital to move forward. Rob is a hedge fund manager, Cindy is the head of their family foundation, and they're both minority owners of the Steelers. So really thinking about how they can take their ties and their time and also their treasure and really move something forward. So we worked with them starting back in 2016. So that's where our journey begins. And as project lead for this initiative, I hit the street going to various conferences, both scientific and patient-led conferences at the NIH, at FDA, ASN, AAKP, and really trying to understand scientifically where some of the gaps were, who are the key players in the space, what's really holding back progress, and what's the state of the science. And in doing so, really came away with a committed group of scientists and patient advocates to come together to assess the current state of the disease space. As you all have heard from several, from Sandeep, from Elena, this is an area that there doesn't really seem to be that much recognition. And that's what I noticed back in 2016. There didn't really seem to be that much known about it with respect to, say, heart disease or diabetes. Go check your cholesterol, go check your blood sugar, but not really much heard about what to do to check your kidneys. And as I was doing my research, I noticed a startling statistic out there that nearly half of patients who end up on dialysis do so in an emergency room setting, which was completely just mind-blowing to me. So we brought together a group of about 30 experts who attended a retreat together with us to really think critically about what the unmet needs were and to prioritize those needs and co-create and think about various ways that one could use philanthropy to do so. And so like I said, we had nearly 30 experts around the table. Dr. Bill Fassel was one, Paul was there with us. And we brought together academicians, transplant surgeons, industry representatives, patients and patient group advocates, and government representatives across multiple disciplines. So we had epidemiologists there, folks who concentrate on dialysis and disease mechanisms. And what we heard from folks around the room was, one, it's not common for all of those different players in the kidney disease space to actually be together at one meeting. So that was really exciting for us. And we covered topics across the gamut, from improving transplant outcomes and dialysis to really getting down to the very basic science and trying to understand disease progression, and then moving up to innovation in clinical trials, and then how we could also innovate in kidney donation. We really, as a center, we tried to take on the full system and try to understand from various angles how one could attack the problem. And after 2 days, we came away with a number of both issues and also possible solutions to achieve that goal. And what the overwhelming thing that we heard was the lack of public awareness of this disease. And so if you look at the cancer space, again, heart disease, diabetes, really when you want to bring funding into a space, you want to raise a national profile, people need to know about the disease. And so that was really the underpinning of all of the discussions that came out at the end. And so in doing so, also we identified solutions that could help move the ball from early detection to, again, innovating in the kidney donation space, and you'll also hear from Bill about things like that. And then the patient and provider education, pilot studies. And so, again, it's really exciting to hear about the things that are going on at HHS and that Sandeep is heading off. So, nice to see those different discussion points actually moving forward in real life. And then, building from there as there's groundswell, understanding patient and donor engagement. So looking at patient-reported outcomes and data integration and wearables. And then from there, thinking about device innovation and patient registries and clinical trials. And finally ending off with clinical trials innovation and biomarker discovery. And so in all of this, what we see is that, again, all of these things require investment, require time, but a lot of that will start to come once we raise the national profile of this disease space. And so as the journey continues, from there, the Citroens really, took all of that information to heart and said, what can we do? What can we, taking on such a massive problem, I understand from several philanthropists that once they start to understand the magnitude of a disease, it all can become very overwhelming. And you start to wonder, what can my little capital source actually go and do? And so, they thought long and hard about their ties, their talent, their time, and thought to themselves, well, there is an opportunity to really raise public awareness. So the Citroens are from the Pittsburgh area. That is their hometown, and as you all know, it's a leader in the transplant space. And so they thought, okay, we can go ahead and launch an organ donation awareness campaign and announce that with the White House efforts. At the time, the Obama White House had really taken up the mantle on organ donation, and they thought, okay, well, we'll announce our effort with that and move forward and plan. And from there, we released the results of all of the year's work and bringing folks together for the broader philanthropic community, for any philanthropist who wants to engage in the kidney donation space as a starting point. one of the other things I heard was that, when I contacted people about this effort, most folks said, well, who are the Citrones? Most people don't give in kidney disease. We don't hear about philanthropists coming to this space. And so, wanting to really lower that barrier for other people who might be interested in the space as a starting point for ideas and solutions. And then from there, as you'll see in the video that I have coming up, they launched the Play It Forward Pittsburgh campaign. It was really remarkable in the city of Pittsburgh, where all 3 of their national teams came together to run a campaign. And I'm sorry.
I was just confused because I'm from Pittsburgh and
Yeah.
I heard nothing about it. I am a season ticket holder for several of the sports.
Interesting. Well, once you see the video, and again, it is something that is still going on. You will be able to see what has been moving forward in the city. And so after this point, once I play the video, I will discuss what ongoing efforts are happening now. So again, I think the video was able to communicate that far more compelling than I could. As an outgrowth of the Play It Forward Pittsburgh campaign, with their eye on the next thing that they could to do to really cement the legacy, was thinking about pediatric transplant outcomes. So from what we understand, no existing group or structure is currently addressing this need. And really where the problem lies is that pediatric transplant outcomes vary widely. So we see rates of serious complication ranging from 2 to 80%, and about 50 children either die waiting or are removed from the transplant wait list because they become too sick. And so what we have heard and researched as contributing factors are, one, the lack of standards. So we've heard that there are few standards, little transparency, and limited quality control in collecting and analyzing outcomes data. And there are also technology deficits where centers are slow to leverage technology to improve those outcomes. And so from that came the Starzl Network for Excellence in Pediatric Transplant. And so what the vision of this network is to really pull together a network of pediatric transplant institutions, academic organizations, and technology innovators who are committed to transparency and continuous improvement to optimize lifelong outcomes after transplantation in children. And then the mission is to unite big data and technology, transplant thought leaders, and patient advocacy to deliver the best possible care and develop new scalable solutions for pediatric transplant. And so I'll end off with saying really that entrepreneurial philanthropy can make a difference. When you engage philanthropists and really get them to think critically about their time and their treasure, their talent and their ties, they can really move the ball forward in a transformative way. And so far what we've seen with the Play It Forward Pittsburgh campaign is that the campaign reach has been upwards of 5 million people. They've registered 1,200 new organ donors in the city of Pittsburgh, and the Citrones have given the Children's Hospital in Pittsburgh a $2 million gift to launch the Starzl Network and have issued a $1 million challenge prize to get things started. And over the course of the past 2 years, we've engaged more than 30 partners all over the city and nationally to really bring this into existence. So thank you.
Thank you so very much. And we're going to have a question and answer session after the panel finishes. But it sounds like there's a lot of opportunity to perhaps work with some of the patient people in the audience to move this even further forward. Thank you so very much. Now our next speaker, has anyone ever had your blood drawn? Okay, I just want to make sure you're awake here, all right? So has anyone here had to wait to find out what their results are, or do you know them right away? Has anyone waited? Of course. We all wait. And if your doctor isn't on the ball, maybe you wait a couple days or longer, unfortunately. So our next speaker is going to tell you about something very fascinating. And interestingly, he is the CEO of FAST. So what do you think that stands for? FAST Diagnosis. And this is a very, very interesting and exciting company that has developed something that I think will be really important to all of us in our diagnosis of kidney disease. So, Mr. Joe Muldoon, you're on.
Thank you, Dr. Schmidt, and thanks to AAKP for inviting me. It's quite an honor. And with a name like Joseph Patrick Muldoon, I'm very pleased to see the strong Irish theme emerging here. I'm going to tell you about what we're working on in Indiana. If I can figure out how to advance my slide. Well, first I'll tell you that Dr. Schmidt and in particular Paul Conway coached me well for this presentation. They said, you know, be clear, be compelling, but above all, Muldoon, please be brief. So I'm setting a timer for myself. Great, thank you. We're working on a first-in-class technology, and what it does is it directly measures kidney function, and it directly and accurately measures blood volume. And we don't have a way to do that today that's clinically viable. We've received a very rare FDA expedited review. They're particularly rare on the device side, which is the group that leads our review. We've received substantial NIH support to date, which has been terrific. I can't say enough good things about NIH and how much they get it. They understand how this could improve patient outcomes. And what it's going to give us the ability is to move past what today are really trailing indicators and pretty crude and insensitive estimates of both kidney function and blood volume. Today, what we have is inadequate. And we've had it for so long, sometimes it's easy to forget how inadequate it is. Today, for kidney function, we track serum creatinine, and we use that in a formula to estimate your glomerular filtration rate or your kidney function. And we have lots of technologies that try to estimate fluid responsiveness to give you a way to kind of approximate what your volume status is. Creatinine is a really poor marker to determine what kidney function is. It's very insensitive, and this is very prevalent in the literature. You could literally publish a book on all the studies that show how bad creatinine is for actually telling you an accurate kidney function. This one is from New England Journal, even back to 2006, and the point it makes is that above 60, creatinine is pretty much completely not useful. Why I think that's a problem is it completely removes the ability for us to catch very early on declines in function, which we believe our technology can do. So this tries to kind of explain the difference in what we're doing and what we have today. So today, what we've seen, particularly in cases where there's an acute loss of function, a very rapid loss of function, is that your serum creatinine that we're tracking will still go up pretty casually and pretty slowly. Patients can have in excess of a 50% loss of function before serum creatinine even bumps outside of a normal range. So then when they take that serum creatinine level and put it in the formula to estimate what kidney function is for this particular patient, it also shows a relatively gradual and slow decline in kidney function. What we've been able to demonstrate with our technology is that's not what's going on. What's going on in these patients is they're actually rapidly losing kidney function, and it's bottoming out pretty quickly. And so there's a day to a 2-day lag, especially in acute kidney injury, where we're not doing anything because we don't know to intervene because we can't tell where somebody is function-wise. Most important slide in my deck is how this might impact patients. For all patients, we think it will enable better dosing of drugs. So as you probably know, a lot of compounds are cleared by your kidneys and or potentially nephrotoxic. Well, today, we overdose and underdose those compounds regularly because we don't know what your renal clearance is. And we can't pick up sensitive or small declines in kidney function because of nephrotoxicity because we can't measure it. We can't measure it that accurately. In chronic kidney disease patients, the biggest one, I think, is a better potential endpoint for clinical trials. We talk about barriers to innovation, and one of the top two consistently in every conversation I've been in is clinical trials are too hard. They're too big, they're too expensive, they're too long, and too many of them have failed. And every time one fails, it makes future sponsors more hesitant to try and fund the next one. Well, one of the reasons is because our outcomes are death and dialysis. Those are the endpoints in clinical trials, and those are really hard endpoints to design a trial around. If you had one as how did it improve or decline your kidney function, that could move clinical trial work forward. It's a better way to detect early onset and potentially stage patients and their treatments for trials. For end-stage renal disease, we've heard a lot about the tremendous discomfort post-dialysis, often related to an abnormal volume level. Well, for the first time, we'll be able to actually tell you what your volume level is in real time. It's about 20 minutes total turnaround time. And more importantly, to avoid or delay the progression on to end-stage renal disease because we can start picking up small declines in function very early on, much earlier than we can do today. For transplant patients, we think it could get more organs in the pool. And we formed this opinion. We did a press release on our expedited review, and my phone started ringing off the hook with CEOs of transplant facilities. And they said, more and more of our donors are cadaver donors, and we don't really have a good history on them, and we can't really tell what the function of their kidneys is. With your technology, we could actually figure out what their function is and we think get more organs in the pool. Also, post-transplant, we think it would give a better monitoring of what's the function of that new organ and probably more importantly, dose those immunosuppressants, which are nephrotoxic to optimize that dose, which is so critically important. Really unique role for AKI patients, which is the ones that mimic the slide I showed you earlier where there's a rapid loss in function that today that we don't pick up. So that could actually be a diagnosis for AKI for the first time and measure the response to therapy. It also has a role in cardiorenal and in sepsis patients that's pretty significant. How the technology works in brief, it's an injectable device combination product. So we've developed a proprietary compound. It's carboxymethylized dextran that we've found a way to label with fluorescent dye. We give patients a very small bolus injection of it, 3 milliliters in solutions. In all our trials, we've done that via an IV catheter. And then we take very small blood draws. With one blood draw at about 20 minutes, we can calculate the patient's blood volume. With 2 more, over 170 minutes, we can calculate the patient's kidney function. And we measure it right now with a validated fluorescence assay, but we're moving that to an HPLC standard and then ultimately bedside point of care. Our clinical history, we've dosed 89 patients to date, and we've given them collectively 97 doses. That's been over 3 clinical studies. We did our first study in Germany, and we've done the most recent 2 in the US. Our patient populations have included healthy volunteers and stage 3 and 4 chronic kidney disease, lots of comorbidities in these patients, including hypertension, diabetes, and COPD. So we're getting increasingly confident in our data that we've got a very safe technology that can work in a variety of disease states. So far, and in our most recent trial, the results were spectacular. No SAEs, extremely accurate, repeatable, and sensitive. Both our primary and secondary endpoints were achieved. We've shared that data with the FDA, and it's gone very well. We think our path for approval is very clear. We have 2 publications in process. One of them recently published in JASN. I encourage you to read it, and we've got another one in process. This is the front page of the JASN one. And what we showed is in this last trial, we compared our measurement to iohexol clearance. I don't know if you're familiar with iohexol clearance. It is believed to be an accurate way to measure kidney function. It's not FDA approved, and it's not used clinically because it's very cumbersome. It takes a long period of time, and you get results back in 2 weeks, which is not quick enough to make a clinical decision, but it's believed to be accurate. So our correlation to iohexol clearance over all ranges of function was 0.996. It's as accurate to iohexol clearance as iohexol clearance is to itself. So we think we've got a very accurate technology. Next steps for us, we've recently completed our end of phase 2 trial. We'll be out in the market raising our Series B capital. We're looking at a potential 50-patient trial in heart failure that the country of Germany has approached us about doing. And we'll get ready for our final approval trial. It'll be a pivotal trial. We think about 200 patients, multi-site, multi-country. We're hoping to get approval by 2021. It's all right. Did I stop on this one? So you've had to look at that slide the whole time, and I didn't even know it. The other ones are better. They have pictures and stuff.
Your time's running out.
Okay.
I'm just kidding.
So that's slide 5. So is there a way to pull up 6? So in sticking with the Irish theme, maybe this is Murphy's Law? But it was advanced in the first 5. Maybe I mixed that up.
Now we've got-
That's all right. Why don't I just wrap it up and we'll, I don't want to get us off schedule. Well, I'll just wrap it up. So you've had to endure listening to me without the benefit of something to actually look at on the slide deck. Oh, and here they are. We've already covered them all. But well, I'll just fast forward to, if you show me which one to push so I don't screw it up again. Yay. We talked about the clinical history. There's our Jason article. I encourage you to go read it. It's pretty cool. We're pretty proud of it. And we think we can get this to patients by 2021. So thanks for your attention.
Thank you very much, very interesting. And you didn't say much about the fluid part, but for patients that are on dialysis, this could be really, really helpful in managing your fluids and monitoring things other than just using the scale, which we all know is so accurate. So, thank you very much, Joe.
Thank you.
All right, our next speaker is Mr. Peter Magg, who comes to us from the company Care Diagnostic, or CareDx. I'm not sure how you say it.
CareDx is good.
CareDx, all right. Now, this company is dedicated to improving the lives of transplant patients, and it does so through a series of non-invasive diagnostics. And I read your website, and I have to say, it is so innovative, you're really going to have to dumb it down for me. So, very interesting, however, that we can maybe diagnose things that we otherwise have difficult time doing, like different types of rejection, a lot easier and faster. Is that a very, very simple introduction, but take it away for us.
I'll make it very, very simple, I promise.
Good.
What a great introduction for the first slide. But before I show you the first slide, I'm very, 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 very humbled of being here on stage. I've been dedicating a long time in my life to transform a specific therapeutic area. And just 2 days ago, I was at the American Transplant Congress. And we had a room full of leaders in transplantation, standing room only. More than 400 clinicians came to our symposium. We had to bring in more chairs. There was standing room only. And for all those clinicians, you don't like to stand. But this is really very, very exciting. I'll dumb it down for you. I'll dumb it down for you, I promise. If I get this innovation going, how do we transform transplantation? We better match patients to organs, and then we do better surveillance after transplantation, because that leads to better patient outcomes. So what does CareDx do? We're building a village to make a difference. You need, as a transplant patient, lifelong support. You know, it's not only one individual, one clinician, one transplant center, it's a whole community that makes that difference. Immune suppression, I believe, needs to be individualized. So personalized medicine is really at the core of what we're doing. The existing surveillance method, and we just heard from Joe, serum creatinine is really not good. It's not good. Who of you ever got a biopsy? Who ever liked getting a biopsy? All right? So think of us as changing serum creatinine on the one hand and trying to alleviate the need for a biopsy. And then organs are limited. So let's make sure that we have them last as long as possible. And CareDx is the organization who makes that difference. Think about an organization. Wouldn't that be great if we can do a full HLA typing in less than an hour? Wouldn't that be great? Well, CareDx, last year we launched the product that can do HLA typing in less than an hour with our pre-transplant Olerup Q-type. It's available now. It's available now. Less than an hour, 11 loci's. Less than an hour, 11 loci's. The other thing, the biggest innovation, and everybody's very excited about, is AlloSure. AlloSure is the latest technology, and I could bore you to death with what I'm very excited about. It takes us 3 days to get an AlloSure result from the blood draw back to the clinician, but it's latest technology. There's a lot of razzle and dazzle that I could bore you with, but at the end of the day, it's very simple. It's the innovation in transplant care. It's the first and only non-invasive test to assess organ health. It enables better management of kidney transplant patients, and AlloSure can accurately determine active rejection. How do we do that? Very simple. The latest technology in diagnostics is DNA testing, sequencing technology. You might have heard about it. It already makes a big change in prenatal care, where you determine the health of the baby even before it's born by testing blood of the mother. So AlloSure is a very simple blood test that detects the DNA of the transplanted organ, tests the organ in the bloodstream of the recipient. If there's a lot of DNA in your blood from the organ, there's a lot of cell injury, so that is a sign of rejection, not good. A lot of DNA from the organ, not good. Little, that's good. That's good. So if you have little DNA floating around, it's even more important than detecting a rejection, because what does that mean? I call them the green light patients, the healthy patient. So when Elena was talking about the picture of the patient, no. Our pictures of patients that we have come in every month are people that we're working with. They're family members, our employees. They're even bankers, they're investors, they are normal human beings. So we want to care for the healthy patient, for the healthy recipient. We do that by better surveillance. So I've been in this field for a very long period of time and still at awe about the variability of care. You go to a transplant center today in the United States, and there are about 265 of them, and I would say there are about 290 different ways of you getting cared. Because some centers, even individual doctors, are having a different approach. So we like to launch, and that's super exciting, in 35 centers for 1,000 patients, a schedule which has been started to be adopted, which is 7 AlloSures in the first year, 4 in the second, and 4 in the next. And to Sandeep earlier, to CMS, I think what I'm most proud of in the last year was that CMS bought in and they reimbursed AlloSure, fully reimbursed. And they partner with us to do that clinical trial. 1,000 patients in 35 centers. And they fully reimburse AlloSure. Because we're the company that's very much dedicated to good clinical trials. I don't expect you to read all those. But what I'm more excited that there's a bright future. Innovation is great, but it can't stop here. So innovation's going forward. So you see us using AlloSure now in pediatrics. You see us using AlloSure in combination with other pharmaceuticals, because we believe in the long-term monitoring of transplant patients. Compliance and adherence is a very big issue, so we're launching about a patient app that will bring all this together. We have patient care coordinators in our companies that help to monitor this longitudinal long-term follow-up. I'm a pharmacist by training. Compliance and adherence is probably the biggest issue. Because if you really ask a patient, are you compliant? Really? I'm supposed to be on a low-dose regimen every morning. I'm not compliant. It's tough. I have big aspiration of what you need to do. These pill boxes is amazing. So let's help to be adherent and compliant. So I talked a lot about transforming transplantation. And of course, I hope that all of you appreciate better matching, better surveillance, better outcome. But organ availability is a big issue. So we were thinking about CareDx, what can we do as a small little company out on the West Coast? We employ 200 people. We do have offices ex-US in Stockholm and Australia. If you ever want to visit, let me know. We'd love to have you. So what does a small company can do? And people like David Conway have been a great inspiration. David Knight. When I spend a lot of time lobbying on behalf of our company and attracting money on the East Coast and in Washington. And we make a very simple policy. Any employee at CareDx that gives an organ will not have negative consequences of making that donation. And when we wrote up this little policy, it caught a lot of people in a storm. So we have launched this a few weeks ago with friends and family. It's going to be a bigger splash. But we'd like to make a contribution to the field of transplantation, to kidney care. And we're humbled because it's for patients like you that we at CareDx get up every morning. Thank you very much.
Well, thank you so very much. And that is a really wonderful way to set the stage for what we hope will be done legislatively. But right now, companies don't do that. And that is a really noble and wonderful thing to do. These type of tests will certainly save me a lot of headache and work. And I think for a lot of you, the anticipation of having to have a biopsy is not a fun one. So we're excited. This will be a really great thing. So next on our agenda, if we have the slides, we have Ms. Kerry Gus. And Ms. Gus is the executive director of Rockwell, which is a company that made a very novel, one-of-a-kind iron replacement therapy that goes right into the dialysate. So how many of you have ever received IV iron? Many of you. And you know how they give it to you at on the machine, or sometimes CKD patients not on the machine get it, or you have to come in if you're not on the hemodialysis machine. You come in and are given it by IV. And that's not as physiologic as just giving it in the dialysate. And that's what this medicine does. So it's pretty exciting as well. And it's called Triferic. I guess you could mispronounce it and say terrific. I like that. How do you like that? That'll be $15. Or more.
That's what we hope.
Yeah,
Yes.
So, as soon as we get these slides up, we will hear from Kerry. How are we doing, team?
Just 1 second.
We may run out of time for questions, but please feel free to stay a few minutes after, because all of these panelists will stay as long as you need to answer any question you have, right? The doors are locked. All right, here we go.
Hello, everybody. I feel like we need to stand up and stretch for a minute, just because I want you to be really excited about my presentation. Yay! My name is Kerry Gus, and I am so honored to be here. As Doctor said, I am an executive director at Rockwell. I really oversee the research and development, a lot of our operations, and have been helping to support Rockwell's efforts to gain the right reimbursement within CMS. So a lot of the conferences that I attend are very clinical, where I'm with other practitioners and doctors, and so to be in this room with actual patients and other innovators is really inspiring. So thank you so much for your attention. I'm also standing up here as an advocate for all kidney patients, which are really near and dear to my heart. I started in this industry about 20 years ago as a renal dietitian and saw firsthand just how impactful and life-changing dialysis can really be. I also have an uncle who was on dialysis for 4 years. It was a very long 4 years. I'm messing up my presentation here, okay? He struggled every day. And being in this industry, what was most shocking and honestly upsetting was how much he didn't know. He didn't know what his labs were. He didn't know really why he would receive certain medication. He knew generally, I'm getting an iron, I'm getting something to help with the iron. But he couldn't answer any of my very inquisitive questions being in the industry. I wrote actually a letter to his doctor, and nothing really changed for him, and it was really hard to see him struggle like that. And he would say, Kerry, that's just how it is in dialysis. And it's actually not. Fortunately, he just celebrated his 1-year anniversary for what seems to be a successful kidney transplant. So that's amazing, yay. And really new therapies become available all the time. And so my ask of you is to pay attention to these innovations and share them with your doctors and nurses. Sometimes we just get so busy in our jobs that we can't look up and see, wow, there's actually new innovative technologies that can really help our patients. I know I drive my doctors nuts all the time, and so that's what I want you to do as well. And so I'm going to talk to you about some other choices you have to help manage your anemia. And I'll be talking about triferic as well. So what is anemia? So who has ever had anemia here? Yep, okay, me too. Anemia occurs when our blood lacks enough of the healthy red blood cells or hemoglobin. And we need to get iron regularly to prevent anemia. Our body uses iron to make hemoglobin, and it's hemoglobin that really enables our red blood cells to carry the oxygen from our lungs to all parts of our body, providing us with energy. And without enough iron, we get anemic. And so for us and for patients, that means we're tired, we feel weak, and we have a lack of energy. Iron is really important for not just physical health, but mental health as well. Iron helps to maintain a healthy immune system. Again, it helps to make hemoglobin. Getting the proper amount of iron ensures that we get oxygen and more energy. We also need erythropoietin and iron together in our bone marrow to help make hemoglobin. When your kidneys fail, you have low levels of erythropoietin. Your restricted diet limits the iron intake, and iron loss occurs with every single dialysis treatment, which we'll talk about in a minute. The inflammation that you have from dialysis and other medications really limits the iron transport to your bone marrow, which is where it needs to be. And anemia then occurs, which makes us really feel yucky. So dialysis patients lose about 5 to 7 milligrams of iron at every dialysis treatment. That's due to needle sticks, blood trapped in the blood lines, in the dialyzer, and just blood draws. That ongoing loss at every dialysis treatment is really the primary driver of your anemia. And so just like we replace calcium and magnesium and potassium, we need to replace that iron really at a maintenance basis. So you really have 3 options. How can we get our 5 to 7 milligrams of iron that we're losing at every treatment? So there's oral iron, which is really not effective for dialysis patients. It has poor absorption, and it has some really bad side effects that none of us like. And then we have IV iron. So when Epogen was approved in the late 80s, early 90s, it couldn't work alone. And so the FDA approved IV iron for repletion therapy. We've been using it in dialysis patients because that's all that's really been available to help replace that 5 to 7 milligrams of iron. It's injected into your blood. The majority of that iron, about 95%, actually goes straight to the liver. All IV irons are encased in a carbohydrate shell. That means it's going straight to the liver to be metabolized, and when it gets there, it gets trapped. And that causes inflammation. A lot of dialysis patients have really high ferritins. That means there's iron trapped in their liver, and sometimes they can't mobilize that, so they're still really anemic. And so now we have dialysate iron, which is triferic. Triferic is mixed in the dialysate at every dialysis treatment, As the patient is dialyzing and they're losing that iron, it's crossing over the dialyzer membrane, binds immediately to something called transferrin, and then is taken directly to the bone marrow. Nothing gets stored in the liver, no increase in ferritins. And so when that patient gets off the dialysis machine, their hemoglobin is essentially unchanged. It's a slow infusion of iron, so we're not giving a big bolus, just a small amount that's lost at every treatment. It actually works in the body just like eating a steak. So when you have lunch next, it's gonna follow the same mechanism of action in your body. What a lot of our patients tell us is they love it because they never know they're receiving it. There isn't a bag hanging, there's no needle. It's just really mixed in the dialysate, no metallic taste at all. So dialysate iron, or triferic, is really the new way to treat anemia, just replacing that small amount of iron that's lost. It helps provide more energy when you have higher hemoglobin levels or maintenance hemoglobin. And it has the largest safety profile of any iron submitted to the FDA, and there's no side effect attributed to it. In our clinical program, it was just the same as placebo, which is the best you can really hope for any drug. And nurses love it because they no longer have to spend the 5 to 8 minutes administering the iron and coming back and pushing the rest. So there's more time for quality patient care. And it's so funny, my colleague over there, Dana, and I were at a clinic who implemented triferic, and the doctor loves it so much, he tells us on a regular basis, this is better than you're telling me it is. And we had an opportunity to talk to the nurse, and she said, oh, we've just hired all these young nurses, and they don't even know what it's like to push IV iron because they're coming up out of school and just using triferic. She said they'll never truly appreciate just how great it is, which is always good to hear. So really the biggest difference, if you view them as two completely separate drugs, triferic is the only FDA-approved iron for iron maintenance, where IV iron is still great for repletion, and that's how we feel it should really be used. Triferic can consistently maintain hemoglobin. With IV iron, they're just giving those large bolus injections. With triferic is used all completely once it enters into your body, nothing's trapped, no increase in ferritin, no increase in inflammation, and we don't have that carbohydrate shell, so in over, probably close to 800,000 doses now, we haven't had a single episode of anaphylaxis, which is great. Actually, not even a single adverse event reported, which is good. So what we ask of you is to take action. Ask your nurse or doctor to get the best iron replacement therapy for you. Educate them. We do our best out in the field to try to educate other nephrologists, but they want to hear from you too. Ask them to learn about it. You can stop by the Rockwell Medical booth and see myself and Dana. Raise your hand. Yay. And we also have a lot of information for patients and healthcare professionals on our triferic.com website. But the real message is you have a choice, and just choose what's best for you. I'm going to end with a patient video. Charlene is someone who's near and dear to my heart. She was a research patient before triferic was approved for years, really in all phases. And once triferic was approved, she was mad at her clinic. Like, I got to have this, and I felt so good during the clinical program, and now I can't have it? Why? And so she drove them crazy, worked relentlessly to make sure that they knew that this is what she wanted. And fortunately, she's great to cross paths with. She is also a big advocate in her hometown in New Jersey. But she wanted to do this video as her little testimonial, and she's just fantastic. So I'm gonna try to find it here. Okay.
My name is Charlene. I am a teacher, a singer, an actress, a choreographer, and of course a mother. The first time I had dialysis was in the hospital. I was going to the doctor, and she said, get her to the hospital now. It was, you have to have a pacemaker, you have to have a catheter. We're talking about a fistula. They just said, oh, we're going to dialysis now. It was instantaneous. It was the first time in my life where there were no choices. That was frightening for me. My life has always been busy. I've never been a still person. I just want to do so much. There's so much out there to be done. And I get depressed when I don't have the energy to do stuff. And when I went on dialysis, I was always tired. Always tired. I'm gonna go home and I'm gonna go to sleep. What kind of life is that? You know, that's no kind of life. And when you don't have the energy, you become very depressed. Because I'm on dialysis, I can't do what I used to do before. And then when you get triferic, it's like opening the window. Suddenly, I can do these things. I'm not falling asleep all the time. I suddenly got an infusion of energy. We have a choice of how we can feel while we're on dialysis. With triferic, I feel like you're getting a little of your life back. I can tell the difference in my body with triferic and without it. Without it isn't a place I want to be ever again. Triferic helped me. It did more than help me. It gave me a better lease on life.
So Charlene was successful in getting triferic implemented in her unit, so now over 300 patients are experiencing what she did, which is great. So thank you so much for your attention.
Thank you very much. That was terrific. And hopefully your enthusiasm can be infused into the patients to go to their doctors and ask about this.
Yes, I know I get very carried away, honestly. I do. I just get really passionate about it. So I hope that was relayed to you and you can at least stop by our booth and learn more or ask your doctor to learn more.
Very good.
Thank you.
All right, so our next speaker is Dr. William Fissel. Dr. Fissel is a very renowned research scientist, nephrologist from Vanderbilt University, and he's going to talk to you about what would be an epic advance. I don't think anyone would disagree with that, an epic advance in the care of patients with kidney disease. Bill?
Great. Thank you so much for your attention. I want to start with a quiz. So I have a pop quiz for you. And the question that I have for you is, in what year did I meet my first patient with kidney disease? And your choices are 2018, 2000, 1987, 1984, or 1966. How many people think it's 2018? That's one. How many people think it's 2000 when I started my nephrology fellowship? That's one. How many think it might be 1987 when I climbed into the back of an orange and white van and started my prior career as an advanced life support paramedic in the streets of Boston? Got a small handful there. How many think it was 1984 when I went to college? There's one. How many think it was 1966? There you go. It was May 1966, the day I was born, because I have kidney disease as well. So for me, there's no us and them. And I'm uncomfortably aware when I have my little white coat and my stethoscope that it is nothing more than chance, nothing more than a coin toss, nothing more than just the whim of the world that I have the white coat and the stethoscope and the person in front of me is staring at the sharp end of a dialysis needle. So I want that to motivate the rest of this discussion. With all due respect, social engineering efforts to expand the pool of donor organs haven't really delivered what we've hoped for. That's why Dr. Riley is pushing forward efforts to expand the pool of donor organs by improving donation rates. And we have to take excellent care of the organs that we get. But at the end of the day, we have 20,000 transplants a year in the United States and 750,000 patients looking at a dialysis machine. What I would like to do, you lied, didn't do it. What I would like to do, I'll use this one. What I would like to do is discard the scarcity model. I would like to use biomedical engineering rather than social engineering to expand the pool of donor organs. My goal is that when a patient is nearing the need for therapy, that they can either get a live donor organ, a deceased donor organ, or a device that will support them and allow them to essentially forget that they have kidney disease. I would like to walk away from the gentle condescension of the physician tapping the patient on the shoulder and saying, oh, don't worry, you'll be able to live a normal life, kind of 2.5 days a week. All right? We've heard already that we have not enjoyed the same technology advances that other areas of medicine have enjoyed. So when I was a paramedic, cardiac rhythm management consisted of me finding somebody dead in the shower. But today, engineering advances have allowed computers to diagnose and treat dangerous heart rhythms. Low-power circuitry lets those computer algorithms be implemented in a little gadget that sits in your chest. So that today, as a patient calling the cardiologist saying, hey doc, the thing zapped me in the shower last night, do I need to come see you? Totally different model. On the other hand, for all of us, we've had in-center dialysis since Culf in 1944, treated a patient with acute kidney injury, and the patient survived. We've had devices for in-center for home dialysis ever since walnut grain Formica was stylish. But what we don't have is a gadget that will deliver enough kidney to a patient in their body so that they can forget about the idea that they're a patient. What do we got to do? How do we get the job done? Well, everybody thinks kidney, they think yellow juice in a porcelain toilet bowl. We have to get rid of wastes, right? You want to eat and get rid of the stuff that you ate. As we heard about water, weight, blood volume, most people weigh about the same from one day to the next because you drink more, you pee more. You eat more salt, you pee more salt. But we all don't get to do that, right? We all have to jump on a scale at the dialysis unit. So a gadget that we designed to put into a patient has to also be able to maintain total body water content about the same from one day to the next. So you're not falling down, cramping, and you're not drowning. Finally, how many people in this room have gotten a phone call from their physician that somewhere in the course of that phone call the word potassium was mentioned? All right, so we also need to be able to regulate ion concentrations in the blood. If we do those three things, we're about 99 to 44% of the way there. All right? How does that happen? We have to do that in a healthy kidney by processing an awful lot of fluid. Normal kidneys churn through 100+ liters a day of filtrate. That's a lot of fluid. I can't carry that with me. I can't put that on a pack on my back. In dialysis, this happens in the water room, right? How many people, show of hands, have been in a dialysis facility where there's water on the floor? All right? So water's generally cheap. We churn through a lot of it during a dialysis session, but the downside is that you're tethered to an electrical outlet and you're tethered to a water supply. Your kidney, the healthy kidney, filters a lot and then uses living cells inside your kidney to take back salt and water, and amino acids, glucose, all the good stuff, and concentrates the bad stuff about 100-fold into the liter or two they should pee in the toilet bowl. So why don't we do the same thing? Why don't we set patients free from the idea that they need to be plugged into a wall outlet and they need to be slopping around in water on the floor? So that's our goal. We've been at this for close to 20 years now. We've been at this for probably 4 times the life expectancy of a patient on dialysis in the United States. I liked what Sandeep Patel said this morning about urgency, because I feel urgency about this. Our strategy is very simple. We're going to build the stuff that we can't grow. Kidneys filters are the thing that breaks in 90% of the kidney disease that results in kidney failure in the US, full stop. I can't grow that. And the moment I grow it, the disease is going to eat it again. So we're going to build that. On the other hand, I don't want to be plugged into a wall outlet. And in fact, the stuff that makes you sick, the stuff that made you feel crummy while you're on dialysis, the stuff that made you feel crummy before you started dialysis, we fancy doctors with our white coats don't know the molecular identities of the things that make you sick in renal failure. Hate to tell you, it's a dirty secret, but we've been dialyzing people for 60 years, and the molecular effectors of the things that cause the residual syndrome and dialyzed renal failure remain unknown. But your kidney knows. I don't have to know it up here. I have to know it back here. So let's use the cells in the kidney that know which molecules have been naughty and which molecules have been nice to grow a Santa Claus membrane instead of waiting for the science discovery process to learn that. So those of you who've seen me before know that we invented a new membrane technology about 18, 19 years ago. It was really not all that exciting in some ways, right? I'm not going to dumb things down for you because you're not dumb. Simple as that, right? All we did was we changed the material that we make the membrane out of. We went from polymer dialyzers to using the same stuff that lets a cell phone know which way is up, using silicon nanotechnology. And we pushed that forward over a number of years, so it came from concept to things that we're now routinely sewing into experimental animals, and that we're pushing for FDA approval to test on patients. On the other hand, where the steep hill has been, I think, is learning how to grow cells in culture. The kidney tubule cells grow like cockroaches. They're great. They'll proliferate like crazy. But what they won't do is make like the 7 dwarfs and hi-ho, off to work we go. Because the way that we culture cells in a dish is fundamentally unlike the way cells live in the human body. And so our challenge has been, over the last couple of years, has been to learn how to abandon legacy habits in cell culture and develop a set of tools that let cells do their job. Very quickly, this is my one science slide, as it were. You can see that there's one bar sticking up, and all the other ones are flat. That bar is the amount of protein that reabsorbs salt in the kidney. The left 2 bars are kind of using conventional cell culture techniques. The bar all the way on the right is using conventional cell culture techniques, and the tall bar is us. So just by changing some of the obvious stuff, and those of you who have seen me speak before know the kind of laundry list of things I'm talking about, are just putting those aside and learning how to let the cells do what they already know how to do. So I was asked to discuss timeline. Predictions are hard, especially about the future. We've been at this for quite a while. We've been incredibly fortunate to have received funding from the National Institutes of Health, and we've received it from the National Institute for Biomedical Imaging and Bioengineering. This is not an NIDDK-funded project. From 2003, when I got my first grant ever, to today, we have been funded by NIBIB. We'd like to try to move forward. So the regulatory process involves safety and efficacy, and I think you've heard that from people here. We really would like to try, we were hoping to get first safety studies into human subjects last fall. We've been engaging in a little bit of a tennis match with the regulatory authorities to try to get them to tell us that we can go ahead. But the applications are in a revision. We've had a lot of conversations. I'm optimistic that we'll be able to go forward with a human safety study. It's going to be brief. It's not like in August we're going to be sewing things into patients and sending them on their way. But we want to do it here in the United States. Your taxpayer dollars paid for the research. You should reap the benefits. So we're going to do these clinical trials here in the United States. If we make it over that hump, we would like to have our first kind of camel's nose under the tent for efficacy studies next year. So this is within the lifetime of a patient who's starting dialysis today. This is not some distant never land off in the future. I started a patient on dialysis earlier this year who had polycystic kidney disease. The gene for PCKD was cloned 20 years ago, in 1998. What do we have to offer that patient after 20 years of basic science research? Okay, this year we have tolvaptan, which was developed for heart failure and hyponatremia. Patients started dialysis on a dialysis machine that was indistinguishable from the dialysis machines that were in use when the gene was cloned. Okay? NIBIB, the bioengineering funding agency, has had the vision to be able to say, let's look at some high-risk, high-gain projects that can move the needle on how we take care of patients today. And that, I think, has the promise of being able to deliver a real clinical result to all of us in a time frame that we can appreciate. We got to move this out of the university to the factory, right? You do not want the device that some guy in a white coat or some woman in a white coat is going to hand to you to have been made in a facility that is completely dependent on how awake the last graduate student was when he cleaned the furnace, okay? So we need funding to move from the university to the factory. The NIH does not fund product development, not NIBIB, not NIDDK, not anybody. So we need a lump of money to get us out of the university, out of the academic environment, and into a scale-up production environment that can get us where we need to be. This is not mission to Mars kind of money. I don't know about you. I don't really want my tax dollars sending spaceships to Mars when I got 12 patients in clinic tomorrow afternoon. This is not rocket science kind of dollars, okay? This is achievable money that could be done with somebody signing a check. That's where we need to be. I would like to see our first human subjects, our first patients, our first us, our first success later this calendar year. It's going to depend on what the approvals process is, and it's going to depend on the stream of dollars that let us move some of the high-cost items in our project from exploration to manufacturing. I think I'm over time. I see people over there looking at their watches anxiously. I'll close with Kolf. Kolf was the first doctor to successfully dialyze a human patient. Others had tried before, and he was certainly not the first person to use hemodialysis. That actually was probably John Jacob Abel at Hopkins 40 years earlier. Kolf was instrumental in the design of the Jarvik heart. And when Kolf actually passed away almost a decade ago, he was working on artificial lung technology. He never quit. He kept going. But I loved his quote that it's nearly impossible to obtain funding for something simple. And everything that I've done, everything I've showed you, is actually conceptually simple. Just change the materials we make the filter out of. Boom. It's more efficient, it's smaller, we can sew it inside somebody. We need the cells to work. Let's just change away from a plastic lab dish to something more like the human body, and hey, presto, the cells do their job. Therefore, over the years, my laboratory has teetered along, always on the rim of bankruptcy, always having too many ideas and too little money to carry them out. All right, thanks for your attention. I appreciate it.
Thank you so very much, Dr. Fissel. Enlightening, disturbing in some ways about the money, and maybe something can come of that through collaborations? If any of you have 25 million out there you'd like to donate, bring it on. But I mean, that does seem like a fairly small amount when you consider how much money we spend on a lot of things. And to those of us in this room,
60 billion?
Yeah.
25
Yeah.
million.
It's
60
pretty
billion,
disturbing.
25 million.
yeah.
You decide.
So our last speaker, last but not least, is a good friend of AAKP's on the Medical Advisory Board, and he brings to you a talk of a new medication that does something quite interesting. How many of you take, like, a lot of medicines? Okay, so if you take 10 medicines and you could take 5 medicines or 3 medicines or even 7 medicines, which would you choose? Duh. And this medicine that he's going to talk to you about does several different things. So it's very exciting to think that you can hit several problems with one medication, and this is not for prime time yet on patients that are already on dialysis, but it's been very successful in people that are with chronic kidney disease, hopefully not, but perhaps headed that way. So
Yeah.
thank you very much, Hector.
Thank you so much. I know that I'm between you and the lunch, so I understand. And I know there are people here who are from Washington and who have been up very late last night. And I am from New York. But I got a text from my son at 2 in the morning. And he says, "Did you watch the Stanley Cup final?" And I says, "Yes." And he says, "Dad, we have hope." And I says, "What do you mean?" He says, "Maybe in our lifetime, Jets will win the Super Bowl." So that was a little bit of an icebreaker, but on a serious note, I'm a nephrologist by training. I was in academia for about 15 years before I moved on to clinical research, but that's not the reason I'm here. The reason I'm here is because I have a very strong family history of like many who are sitting here, and when I was in academia, I used to tell my patients that you're not unique. You know, we have so many people who have kidney disease. If you go to a restaurant, you will find a person who is sitting next to you may have a kidney disease, and the person who is sitting next to that person may have also kidney disease. He doesn't know it. So there is a lot of kidney disease out there, and unfortunately, I am not special. So I have a very strong family history. One of my uncles who stayed on dialysis was on dialysis for 4 years. Unfortunately, unlike Kerry's uncle, passed away without able to get the kidney transplant. And one of my aunts just started dialysis 2 months ago. And I live in New York, work in Miami. I've never traveled to Atlanta as many times as I did in the last 2 months because everything happens and I have to jump and see my aunt who is on dialysis. So there is a reason I'm here, and there is a purpose for us to all be here. So my passion towards AAKP is the same as yours. Is we want to make patients' voice heard. We want to have more patients engaged. So without further ado, I'm going to talk a little bit. This is the disclaimer. Again, this is an educational discussion. So, when we talk about vitamin D, we talk about, people who may be living on the South Pole or the North Pole. But, to be honest with you, that is not true. If you look at this slide, it will tell you that vitamin D deficiency is prevalent all across the board. If you're on the South Pole, North Pole, or in the middle, this blue thing you see where we don't have any numbers attached to it doesn't mean that the vitamin D deficiency doesn't occur there. It's just we don't have good data in that area or in those geographic locations. So it's a very, very prevalent issue. But this is something which you go to the doctor. I'm sure everyone in this room go to have a doctor's visit, and I think this is a very common thing that people check the vitamin D levels, right? When I talk to, not the patients, when I talk to some of my colleagues and I says, okay, tell me, vitamin Ds, and, they really don't have the idea or clarity of that all vitamin Ds are not the same. So you talk about the vitamin D2 and D3 and all those, and people really don't have the that clear understanding, even though we go to the doctor and doctors check our levels and put us on some medications, and we have no idea what is going on. So basically, there are two types of vitamin Ds, which we call the pro vitamin Ds, vitamin D3 and D2. So D3 comes from the skin under the influence of the ultraviolet radiation. The cholesterol gets converted into cholecalciferol, and that's one of the animal sources of vitamin D. On the other hand, we have what you call ergocalciferol, which comes from the diet. It's a plant-based vitamin, and these are the two vitamins actually, D2 and D3, which we take in a lot of nutritional supplements, multivitamins, these are the ones who are present there. But these are not the actual active hormones. These have to be activated to the second part of the pathway, which is the 25-hydroxylation, which occurs in the liver. And then this 25-hydroxylation, after it occurs in the liver, it has to go to the kidney to get another hydroxylation, which is the 125 hydroxylation, and that occurs in the kidney. And that's what makes you the active hormone. So 125-D3 is the active hormone, and we're going to talk a little bit about this. But I think people have a little bit of conceptual misunderstanding in this pathway. So when you go to check the level, you're checking the level of 25-hydroxy-D. You're not actually routinely checking the vitamin D3 level or vitamin D2 level. So just wanted to make sure we understand and are on the same page. Why are we talking about this? One of the reasons we are talking about this because one of the things which we want to keep in the normal range is the serum calcium. And serum calcium, low or high, is bad. Low calcium can cause tetany, cardiopedal spasms. High calcium is associated with arrhythmias, kidney failure, and a lot of other things. So what happens when the serum level of calcium goes down? In response to the low serum calcium, parathyroid hormone gets secreted, and this parathyroid hormone has multiple functions. It absorbs it retains the calcium from the kidney, absorbs the calcium from the gut, and also activate this 125-hydroxy-D3, this active vitamin D. And the main purpose of this active vitamin D3 is to increase the absorption of calcium and phosphorus from the gut. And that results in the normalization of serum calcium. But this is, as you know, is one of the big problems in patients with chronic kidney disease. So I'm going to just quickly go over this cartoon, which just to illustrate my point. I already talked about the vitamin D3, that is the one which is coming from the skin, or vitamin D2, which is coming from the diet. It has to go to the liver, but what happens is most of that, either the D2 or D3, does not get to the liver because it's a very lipophilic, fat-loving particle, so it ends up mostly in the adipose tissue rather than actually getting to the liver. So one of the reasons why people don't see increases in the level of vitamin 25-hydroxy-D after they're taking the nutritional supplements for a long time is because it may not be getting to the liver. That's where it has to go. And one of the reasons one of the things which causes vitamin D deficiency is obesity. There's a lot of data which suggests that if your BMI is greater than 30, then you are at a high risk of vitamin D deficiency. And this is the rationale that a lot of vitamin D which you take from your nutrition actually end up in the adipose tissue, not able to get to the liver. Now, the second one, which is we take calcitriol, which is the active hormone, which actually it is all preformed active analog. It actually does not require any conversion from D3 to 25-hydroxy, D3 to 125. You are giving that pill or getting the injection in a routine form. But the downside of those is that, these are pharmacologic drugs. They increase the level of exogenous 125 very high, resulting in a high surge of calcium and phosphorus absorption from the gut. And body doesn't like these pharmacologic interventions. There is a negative and the positive feedback system in the body, to regulate a lot of things which go on in the body. So this negative loop kicks in, which destroys or breaks down this 125, but it also breaks down the 25-hydroxy. So the people who are taking the active analogs, if you want to check your vitamin D levels, which we usually measure in the blood, you will find that most of the time they are going to be low because this active hormone is kicking the negative loop, feedback loop, which breaks down this 25-hydroxy-D as well. So let's quickly talk about the secondary hyperparathyroidism. I think in this room, I think almost everybody knows what the secondary hyperparathyroidism is. Secondary hyperparathyroidism, very prevalent. 40 to 82% of the patients with CKD3 and 4 have this disease. It is manifested by increase in the parathyroid hormone level and low vitamin D levels. And untreated SHPT can actually be detrimental. If you don't treat your parathyroid hormone levels, the gland, I will show you a slide, the gland gets bigger and bigger and bigger, and you get to a point where no matter how you treat it, no matter what medications you treat it, it doesn't regress. That's what happened to my uncle. He had to have a parathyroidectomy, and I always blame myself that I wasn't proactive in addressing those before he get to that stage. So it's a really major issue. Now, I always give this example of chronic kidney disease and the progression of chronic kidney disease to kangaroo. And I don't have the time. As I said, you're waiting for your lunch, so I'm not going to quiz how many people know one of the characteristics of kangaroo, but one of the biggest characteristics of kangaroo is that it doesn't have a reverse gear. So it can only go forward, it doesn't go backward. And this is what happens with the chronic kidney disease. It's a progressive disease. And I hope at one point we are able to come up with all these interventions we are talking here where we can actually reverse that course. So as the kidney disease progresses, if you don't treat the vitamin D insufficiency and you don't treat the secondary hyperparathyroidism, the vitamin D level continues to go down, PTH level continues to go up, and that is what is manifesting it on this slide. As the kidney function decreases, your vitamin D level decreases and your PTH rises. And I had mentioned about this. This starts with a normal gland, and as the kidney disease worsens, the SHPT worsens, and you get to a point where your gland is completely transformed into a nodule which is autonomously secreting parathyroid hormone. So at this point when you get there, no matter how you treat it, either you treat it with the active analogs, you treat it with calciumomatics, nothing probably is going to work, and you're going to continue to suffer from the consequences of this terrible disease. SHPT actually and vitamin D deficiency is not a benign thing. It actually causes lives. This is a slide which shows that vitamin D insufficiency is implicated or associated with worsening in kidney function. And this is another slide which actually shows that people who have significant SHPT have a higher mortality. This is not a news. I'm not telling you anything new. And you know the consequences of vitamin D insufficiency and SHPT, not only the bones, which used to be a big area of interest long time ago, but a lot of people now believe that it's more than just the bones, and that's why the disease has changed the name, and we call it CKD-MBD, which is the mineral and bone disorder. So there is a lot more, and I think in next few years, and I think some of the people on the panel in the future will be talking about how you slow down the progression of vascular calcification, and if we can regress the already formed the vascular calcification, because I think that's one of the biggest reasons why our patients who are with CKD actually develop heart failure more often than the patients with the normal patients. So this was a slide which was recently showed at the NKF meeting, and I borrowed the slide from Tamara Isakova. And what she talks about here is that if you get to the end-stage kidney disease, what you're doing is you are abating the severity. You are not doing any prevention. By that time you get to the end-stage kidney disease, you are picking up the pieces of something what should have done before, and you're just just trying to abate the severity. One of the things which recently came out, a study called CRIC study, which I hope some of you get the luxury to read. Basically, it tells you that we spend almost 9 years in CKD3, almost 4 years in CKD2, CKD4, and 7 months in CKD5. So just think about this. If you really want to make an impact, how you don't get the patients to dialysis, that's the time to intervene. You cannot wait until the patient gets to dialysis. And I think that's one of the biggest things which we have to think of. And this brings me to a new molecule which we developed called extended release calcifediol. It's a pill you take at night, and it's a 25-hydroxy-D. So it does not require conversion from the D3 or D2 in the liver. It's already preformed 25-hydroxy-D. And what it does is it's very gradually, slowly over the period of days to months, actually get into the steady state situation. It takes about 3 months for this drug to get into the steady state situation. So it works like in a normal physiologic manner. It's not doing anything to alter your pharmacology. So there is no red flags going on in the body that the body has to kick in to do some work. It's working in a very normal physiologic manner. So that's what it does. It gets from the liver to the kidney to very slowly increase the 125-hydroxy-D, making the active hormone. And that's one of the reasons why it doesn't increase your serum calcium too much. It doesn't increase the serum phosphorus, and it does not increase the FGF23. That's another thing which is going to be you will hear a little bit more in next few years about that. This is some data. It's very nicely and in a slow, gradual manner increase your vitamin D levels. This is the placebo. This is the vitamin D levels. At the end of 26 weeks, the mean vitamin D level was 69. It was continued to another 6 months. At the end of 12 months, the mean vitamin D level was 79. I don't think you will ever hear this level of vitamin D with any other medication. That's not the kicker. The kicker is this. Nicely decrease the parathyroid hormone level. But don't pay the attention. FDA gives the indication to have this drug out in the market to treat SHPT and vitamin D insufficiency. So you know that it's going to decrease the PTH. I'm more interested in this line. This is only 26 weeks, right? 90% of the patients who were not treated had an increase in their PTH levels. KDIGO guidelines suggest that patients who have abnormal PTH and the PTH is progressing are the ones who you should intervene, either a dietary modification, watching their phosphorus, checking the vitamin D. So this is something which is very interesting, and is when the patients who were switched to extended release calcifediol, you could see that the PTH level continues to go down, and the people who were in placebo also came down nicely. And this is another thing which Dr. Schmidt pointed out, is that almost no significant increase in the calcium and phosphoruses. So you have a drug which is increasing your vitamin D levels, there's a drug which is decreasing your PTH levels without significantly increasing in your calcium and phosphorus. So again, this is some data which actually tells you that 0.2 milligram per deciliter increase in calcium in the Rayaldi group and 0.1 milligram per deciliter increase in the control group. This is something I want you to pay attention, and not just for this drug, but any drug. It is really, really critical to look at the adverse event profile. When you go to a doctor, especially when somebody is putting you on a new medication, it's really incumbent upon you to ask your physician the risks, the benefits, side effects, because Alana said very nicely that you are giving your lives in the hands of doctors, and you think we are going to do a good job, right? But there is also an obligation on your part to make sure that you ask the right question. I give you a simple example. My mom had high blood pressure. Her blood pressure wasn't controlled. The doctor put her on a new medication. Next day, she called me and she says, my legs are swollen. She had the luxury to pick up the phone and call the son, and I could tell that she was on a dihydropyridine calcium channel blocker. But I think that's one of the things which you take away. Don't take anything from here. If you take one thing away, is that you really need to get engaged in your care, because you are the only defense. You know, there are people who are going to try to do things to you, and it's your job to make sure that you know what people are doing to you. So I'm going to stop here, and I don't know if we will have the time here, but I will be available today and tomorrow. If you have any questions, feel free to connect with me. Thank you again.
Thank you. Thank you so much, Ekta. So, who said there's no innovation in kidney disease? So very, very enlightening and very exciting. Thank you so very much. And please join me in thanking our panelists. So we'll take about maybe 2 questions before we adjourn for lunch. And for those of you adjourning before the questions, lunch will be on the mezzanine level, right above the lobby in the main part of the hotel. So are there any questions? Yes.
Dr. Fussell, you said you needed $25 million in order to go through the clinical trials. Over what period of time, and are you looking at Kidney X for some of that funding?
Well, so the time period for proposed clinical trial itself will actually be relatively quick. But what we have, the process of moving our technology out of the academic laboratory to a commercial foundry is where the big bite in the money is. So I can go to South Korea and get 1,000 of my silicon membranes made in 30 seconds, but I have to convince them that it's financially worth it to them to take their entire assembly line away from making iPhones and Samsungs to making stuff for us. So that's the big financial bite. Then the trials themselves will go pretty quick. So I would imagine that if the rate limiting step of funding for what we're trying to do got lifted off of us, and we could get boluses of money that would let us do things that are not hypothesis testing, basic science, NIH flavored research, but instead moving a concept into a cost-effective treatment for a patient, we'd be under the 5-year time frame to get from where we are today to having a coverage decision from CMS.
Thank you. Any other questions? All right. We'll take this one here, and then Elena, and then we'll adjourn.
I'm surprised that none of the speakers give a handout. None of these things is going to remain in our head. So you should have given some way in which we can carry this.
I think the question was, could there be a handout provided with some of this information for the participants at the meeting?
We have a lot of information we can give you at the Rockwell Medical booth in the exhibit hall.
All right. And Elena?
One, I have a selfish question. Are you guys presenting at ASN? Because this is wonderful information. And I've been in the business since God was a baby. And it's really cool to hear this. I was telling Paul, the innovation I remember is when we got rid of glass syringes and we stopped autoclaving everything. You know, I did the first peritoneal dialysis at what's now Johns Hopkins Bayview. So it's pretty cool to see. And chasing the money down, God bless you. I mean, but is this information, and how is this information going to be shared with this public? You know, not just through medical journals, which a lot of our patients don't have access to, but how are you all getting the word out to people like myself, policymakers, you know, and patients?
Well, I'll take a crack at that. I mean, I applaud AAKP for being progressive enough to do something like this. I don't think any similar platform exists at ASN. And ASN used to have like an innovators row where you could exhibit, but there was a 30-foot curtain around you and you're in the back of the exhibit hall. So the people that knew you were there would find you, but not a lot of new people would. So I don't think there's a similar platform.
I do think, quite frankly, we need to do a better job of that. But as you said, AAKP is the leader in doing that. So when we published our Giving Smarter Guide last year, Paul and team cross-posted that on their website, and it's freely available for download on our website. But we certainly need to do better in getting it to policymakers and getting it in front of people who can make decisions and have the funding to move mountains.
Have you all partnered like with NKF, or, to try, because I keep thinking of patient organization. When we say, and you're right, one of the things that is my personal mission as a nurse, not as a CMS employee, is to teach patients that they have choice. Because so many now, and I'm just as guilty of it when I'm in a patient role, that I don't always ask the right questions, but to be able to share this information so the patients know what to ask and know what innovations are out there, that not only does it give better care, but it gives hope. And I think that's the biggest thing, is to instill hope in letting, and what you've done with your employees. I mean, it's like, my God, can I transfer now? I just think that it's just incredible. And I really would like to see if there's any way that we can partner, like the Quality Net meeting that we have every year as CMS, which is a free national meeting that is a quality, process innovation meeting, whether or not that would be a good platform to do some of this stuff, because we need to get the word out there. You guys are doing the work. We need to share the info.
Well, thank you so much. I mean, it's very encouraging. I think we're just in the beginning. If you think about all the analysis and innovation that's actually happening in the field, I do think it takes a village. And the transformational approach, I think, is having patience. And we heard that, I think, loud and clear this morning, right from the beginning in understanding what these technologies can mean. For AlloSure, it really means that we are surprised that already 50 transplant centers have started to adopt the technology, and we're just in the rollout. Can I imagine that, in 2 years' time, a transplant patient will come to the doctor's office and say, hey, I would like to know my AlloSure? Absolutely. Yeah. Today, all of you know your serum creatinines. In 2 years' time, you might know your AlloSure. Yeah.
All right, well, thank you very much, everyone. Thank you very much, our esteemed panelists. And stay tuned for more excitement after lunch.